This Week in Virology, the podcast about viruses, the kind that make you sick.
From Microbe TV, this is TWiV, This Week in Virology, episode 897, recorded on May 3rd, 2022. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Austin, Texas, Rich Condit.
Hello there, Vincent.
How's it down there in Austin? You have good weather?
It is 87 degrees, mostly cloudy, so it would be meh, except the temperature's okay. It's just a little gloomy. But it's a beautiful spring here, and the garden is in better shape than it's ever been in. It's glorious. So it's all good.
Also joining us from Ann Arbor, Michigan, Kathy Spindler.
Hi, everybody. Here it's 52 degrees Fahrenheit. I just lost what it is, Celsius, 11, and it's raining.
Yeah, it's chilly here, 16C and partly cloudy. Also joining us from Madison, New Jersey, Brian Barker.
Hi, 65 Celsius, partly cloudy. 65 Fahrenheit, 18 Celsius, partly cloudy, and an okay day to be inside.
It was sunny when I came down. I went to Columbia this morning. I'm cleaning out the lab. I'm throwing out the minus 70 stuff.
Wow, Vincent, this is momentous.
I have to show you this. I mean, we're filling the red bags with vials of viruses. But at minus 70, another thing is, when Saul Silverstein left, he never cleaned out his minus 70. So I'm cleaning out his bloody herpes viruses. Well, not that matters. It just, you dump them in the garbage because he said, throw them out. I don't want them. Antibodies, mutants, constructs. It's such a shame that no one wants any of this, but I'm cleaning up, but I'm giving a good chunk of the collection to Amy and she'll take it with her. So that's all good. I have no problems throwing out stuff. No problems whatsoever. All right. Today we have,
Before we do that, I'm sorry, I didn't get this PSA in.
Oh, sorry.
Remind people there's early bird registration for ASV. It ends this Friday. Costs of all the registrations go up $75 if you haven't registered by the end of Friday. And then the following Friday is the deadline for ASV CARES applications if you want to ask for some money to help pay for a caregiver. And for the Kitty Corp third-party daycare that's going to be on site at the meeting in Madison. So a couple of deadlines. This Friday for registration, the following Friday for ASV Cares and Kitty Corp daycare.
For a minute, I said, did I register? And I said, yes, I did register. Hotel.
I didn't. Register. I'm glad to know this. I will take care of that.
Better do it, dude.
I was just wondering if I could volunteer to work at Kitty Care. I probably have to be vetted or something like that. I'd probably fail a background check.
You would pass the background check, but probably have to do that check in time. Yeah.
Brienne, did you register already?
I have registered, as have some members of my lab. We're all set.
Right. Very good. All right. We have a snippet in a paper for you today. The first is the snippet in the Journal of Infectious Diseases. It's a perspective. Thought we would chat about it. The concept of classical herd immunity may not apply to COVID-19. It's written by David Morins, Gregory Folkers, and Anthony Fauci from the National Institute of Allergy and Infectious Diseases. And I wanted to talk about it because we haven't really that much. We kind of casually talk about it and what it means. So they define herd immunity as commonly understood, they write. I don't know how common it is, but maybe amongst scientists. Herd immunity thresholds are reached when a sufficient proportion of the population is vaccinated or has recovered from natural infection, such that community circulation is reduced below the level of significant public health threat. And they say this threshold has been met with polio and measles circulation in the U.S. I don't know about measles, but it's a little complicated with polio. And I just want to mention it briefly that we probably have vaccine virus circulation. and maybe some of the wild type as well, but we just don't look that much. So I'm not sure, but let's assume that's correct.
I like
ahead,
they have
Sabrina.
this little definition as well in a box. So they have this
Yeah.
box 1 definition. And I really appreciated the way that they had it phrased there. And I'm planning to take some parts of this for teaching purposes. But they mentioned here the threshold is a proportion of the population with immunity above which transmission is largely prevented. So we've had all of those discussions about disease and infection and things, and they specifically mention it's about stopping transmission here. And I did like that.
The other thing is it says the transmission is largely prevented except for sporadic outbreaks in under-vaccinated or otherwise incompletely protected subsets of individuals, like Disneyland.
Yes.
They don't say like Disneyland. I added that.
Right.
But yeah.
Right. Yeah. Okay. I mean, the reason I read the one in the text was I kind of liked it. I liked community circulation is reduced below the level of significant public health threat.
Yeah,
rather than preventing transmission, largely preventing transmission. I think that's too specific, but that's just my.
Well, it actually strikes me that those two definitions are not completely consistent with each other.
I guess what I know is that I think that if you look in a lot of textbooks, you see things like herd immunity is where we stop infection. And I like the fact that neither of these talks about stopping infection.
Right.
Yeah, that's good.
This is timely too, because, herd immunity everybody was talking about it in the early stages of the pandemic. And recently nobody's been talking about it at all. Okay. And yet, although we're going to talk about the nuances of the term herd immunity, here in the Austin area, there's really minimal disease burden and has been for quite a while. And I presume that's because there's a fairly high level of immunity in the community. But whether or not you can call that herd immunity based on whatever your definition is another question. And that's what we'll talk about.
Yeah, I mean, you could say this is the classic definition, but then there's herd immunity against disease, right? If you have high population immunity, maybe you have a lot of asymptomatic infection. Depends how much you're checking, right? So now we have the ability to test a lot of people, but for measles and polio, we have never really done that. So I'm not sure we would know. Kind of use disease as the metric for this circulation, right? I'm not sure that's right. Anyway, the argument is that COVID is different. SARS-CoV-2 is different. So herd immunity, the classic herd immunity, classic rock may not apply to. I think that's funny, classic herd immunity. And the differences between polio and measles and SARS-CoV-2, they say phenotypic stability. So they're relatively stable antigenically. They do change, but not enough to impact immunity. We've been using
polio and measles.
Polio and measles. We've been using the same vaccines since the 50s and 60s, right? And they don't have to be updated at all. And then they also say polio virus and measles virus can elicit long-term protective immunity, at least as far as we know, right? And so they say chasing herd immunity for COVID may be an elusive goal, which is an interesting way to put it. Then they go into a little bit of history, which I appreciated. They said these ideas about herd immunity actually arose before the microbiology era. And they say the 1720 European and colonial introduction of smallpox inoculation made it clear that natural infection and inoculation protected against infection and reinfection. And widespread use of smallpox vaccine prevented or limited epidemics, including during the Revolutionary War. And that's this is what he calls herd immunity, which eventually led to the idea that eradication could happen. And then they go on to say around 1807, it was a crucial observation about measles. They noted that in cities and large towns, measles occurred in cycles, regular cycles, and they varied in length from 2 to 3 years to 6 to 7 years. And different cities had different characteristic cycles. There were longer cycles in towns and smaller cities, and shorter cycles, more common in larger, crowded cities with high birth rates. And so that was a mystery until we made some advances in microbiology, immunology, epidemiology, and they said that we realized that measles infection elicits long-term protection until you get enough children born who are susceptible, and then you have an outbreak. And if you're in a big city with high birth rate, the cycle is going to be shorter. Another study they cite was this 1904 to 1907 measles outbreaks in British primary schools. This is interesting. They said classroom epidemics occurred when fewer than 70% of the children were immune and were prevented when you had immunity around 85%. So I think that was the first time that you could actually put numbers on this idea about herd immunity. What number of immune people, what percentage of immune people did you need to stop outbreaks? They said, though, even though we could come up with these numbers, they didn't have a name. And the term herd immunity actually came from veterinarians during World War I.
I found that amusing.
Isn't that interesting? Actually, there's a reference, number 13, contagious abortion of cattle, 1917.
There's so many references in this that I want to go back and try to read.
And they're so old.
Yes.
They are.
There's one in German, but 1917. They're just impressively old.
So then that terminology became adopted, and that's where we are today. But they thought that herd immunity, which they studied in animals, would apply to people right away, right? But they say numerous variables affected herd immunity thresholds, including, differences among diseases and hosts, transmission modes, completeness of immunity, crowding, population movement, small pockets of non-immune persons, and they all interact to make herd immunity situation specific. And it can be altered by small changes in some of these variables, right? Population density. So they end up saying herd immunity is a useful concept, but really inadequate in real-world situations. And this is quite clear, right? Because let's say you have measles and you're at a population level of immunity, and then you have an outbreak. Well, people will say, well, what happened to herd immunity? Well, that was in a population of people who, for example, were not vaccinated, and they happened to band together, right? So it's important to view this as a concept, I think, and it's not going to apply to every situation. They say that herd immunity concepts over the past 70 years have been repeatedly questioned. For example, influenza, there is inadequacy or limited durability of immunity after vaccination and infection. There's antigenic drift. And as a consequence, and small numbers of unvaccinated individuals, we never attain herd immunity thresholds with influenza. Measles, same thing. We controlled measles by mass vaccination, but there were always outbreaks, and this is caused by importation of virus from elsewhere. So even though you have nationwide 85% immunity, there are little enclaves of less immunity, and if you introduce the virus into them, boom, you have an outbreak. They even note, Rich, with smallpox eradication, they had to contend with repeated outbreaks among unvaccinated individuals until the last natural case in 1978.
Yeah, I think smallpox is probably the only case where herd immunity ultimately has resulted in the disappearance of a virus. Yeah. But that's, though I suppose maybe rinderpest fits into that since it's another one that's been eradicated. Yeah. But smallpox, at least, is kind of a special case. There are several characteristics that make that particularly low-hanging fruit.
Isn't one of them that there are very few asymptomatic infections? Right?
Yep, that's one. Very
So
few asymptomatic infections, lifelong immunity,
right?
No significant variation, and no animal reservoir.
And also no stigma against people saying, no, I don't want to be vaccinated, or people being afraid of getting smallpox.
The other thing that I really love about this doesn't really figure into herd immunity, but it figures into the eradication campaign, is that you can tell by looking at somebody whether they've been either vaccinated or had the infection, because it leaves a scar.
Yeah, so in a way, it's very different, because during the end of the eradication, they would identify a person with smallpox quite clear, and then do ring vaccination around them, right?
Right.
So, and then if another one popped up, you would see it. It wasn't like there were any asymptomatic infections to have an issue with them. And they contrast that here. They say, respiratory virus, influenza, respiratory syncytial virus are very different. There's asymptomatic transmission, there's incomplete protection, there's antigenic variation, and that includes SARS-CoV-2. They call immunity against SARS-COV-2 a fluid concept.
Are you saying immunology is not all one thing?
It's not black and white or white, ranging from complete and durable to immunity that protects against severe disease but does not prevent reinfection and onward transmission. Yeah, right. It's not all the same. So they say classical herd immunity leading to disease eradication or elimination is almost certainly unattainable with SARS-CoV-2.
I think their title is misleading. The concept of classical herd immunity may not apply to COVID-19. And I think really at the end of their second paragraph, what they're really trying to say is that controlling COVID-19 by increasing herd immunity may be an elusive goal, may not apply, may not be possible. So it's more like the concept of controlling COVID-19 by increasing herd immunity may not apply.
Good point.
Yeah,
good point. Herd immunity does apply, but it doesn't work.
It's not working, yes, because of all these reasons,
Controlling this by...
Good point.
But in that case, how would you define herd immunity if you don't have the controlling as part of the definition?
Yeah, I think that's right. It doesn't apply, right? Because you can't control it for the reasons that we've talked about.
Yeah, and then at the very last paragraph, living with COVID-19 is best considered not as reaching a numerical threshold of immunity, but as optimizing population protection without prohibitive restrictions on our daily lives.
I agree with that.
So we no longer need the elusive concept of herd immunity as an aspirational goal. COVID-19 control is already within our grasp.
Yeah, they talk about influenza, which ever since 1918, its descendants are still causing seasonal outbreaks and occasional pandemics. And they say that probably we're going to have a similar situation with SARS-CoV-2. We can do community control with vaccines and diagnostic tests, and we're there already.
Yeah.
But we certainly could. They say we should work on more broadly protective vaccines. But right now we have them. We have the ability to control this. And basically, this is what it is at the moment.
To me, this goes back once again to one of the various earliest episodes with Ralph Barrick, where his prediction was that ultimately this would become another human coronavirus. And, under the best circumstances of immunization via either vaccination or infection, there's going to be virus circulating sufficiently so that, if you happen to not be immune, you're a target. And even if you are immune, there's a reasonable chance that you're going to pick up an infection and get at least a few symptoms, if not more than a few. So, as we've said, it's not going away.
Yeah, I think that the one thing that they underestimate here, or that they don't really talk about, is they don't talk about the importance of herd immunity as a general concept in talking to the population. Because when I think about herd immunity, I don't always think of a number and control. I think about the idea that vaccines have population level effects in addition to individual effects. And that we get something more than kind of the sum of vaccinating 52 people. By vaccinating 52 people, we also get an effect on spread in the population. And so I think when I think about herd immunity, I would broadly want to talk about it as a population level effect. And that's still true. It's just that we're not going to get to that control part like Kathy was mentioning.
Right.
But we shouldn't sort of say, well, I'm only getting a vaccine for myself. You still are helping protect others when you're getting vaccinated.
So the kinds of studies they talk about with measles, where you see outbreaks when you have fewer than a certain percentage of people immune, and then much fewer outbreaks when you have are we in a position to start getting those data for COVID now? Right? In this school, the SARS-CoV-2 immunity dropped and we had an outbreak. I mean, I'm not aware of any, but it seems to me that we could, right?
Yeah.
And that might be interesting. But then, you know, what are you going to define as immunity, right? That's the problem. Exactly. Because mostly I think they're talking about serological assays, right? In this perspective, antibody testing. They're certainly not talking about non-neutralizing antibodies or T cells. But I think if you have the wrong immune component, you're not going to get the right information. I think
Go ahead.
I was just going to say, for measles and polio, we had the right antibodies were correct, right? And we're just not sure that's right here. At least I'm not.
Yeah, they make a point somewhere about systemic circulation of the virus and that differing between groups of viruses. And I really thought it was interesting that they mentioned it.
Yeah, so that idea is that smallpox, measles, rubella, you know, they have a viremia phase, and they also induce robust, long-lasting immunity, and maybe that's correlated, right? Where these mucosal viruses do not. Well, I mean, not mucosal, but
respiratory.
Respiratory, yeah.
I was just wondering if you think it makes a difference to talk about herd immunity versus herd immunity thresholds. Is that just slicing things too thin, or because like in their two definition boxes, they're talking about herd immunity thresholds.
Yeah, that's true.
And in their title, they talk about just classic herd immunity. I don't know. Maybe, again, it's just too semantic or something.
I kind of like that distinction, Kathy, because I think that gets you able to say population immunity is a thing, but there isn't a magic number.
Well, there are two things, right? Herd immunity is the concept. And then if you want to put a number on it, then you put threshold. So for measles, it's 85% immunity. That's the threshold. You go below it, you have outbreaks. That's how I would see it.
Except that I thought it was 90% for measles, or even higher.
Yeah, that's right. Yeah, you're right. In my class, I have a 90-something% number. Yeah. You're right. Yeah. Well, though maybe, but of course, that could depend on the population, right? The reference 12. Yeah, it was that. But I think that's consistent with this fluid concept of herd immunity as well.
So an important couple of messages are that the concept does not apply equally to all viruses. It kind of, depends on what critter you're talking about. And I would also want to make sure that people understand from this that, one could be misled by the title in thinking that SARS-CoV-2 is extra special at not applying or evading this concept. And that's not the case either. We have other viruses for which the concept, the concept applies differently to different viruses. And SARS-CoV-2 is not like off the charts or extremely different, okay? But it's not as respondent to the concept of herd immunity as polio or measles. We're going to live with it.
So Rich, in there's a number for smallpox. I don't remember what the threshold is. Okay.
Neither do I.
But I presume the way they did serological assays looking for antibodies, and then they said, okay, when we get above this certain percentage, we see a decrease in disease, smallpox, the disease, right? So you make a correlation between the two.
I'll bet you there was not necessarily serology involved. Like I said, you can tell who's vaccinated and who's not. Okay. So, and plus we had good vaccination records. There was mandatory vaccination in lots of places and rigorous vaccination records. You couldn't cross borders without your vaccination certificate. You could tell whether or not you're vaccinated. So my guess is that in that I'm guessing, I'm making this up, but it's hard for me to imagine that there would be a lot of serological assays. It probably has to do with the amount of disease compared to the fraction of the population that had either been infected or immunized, because that's an easy number to get, relatively easy.
So the main point here is that there are viruses for which classical herd immunity likely doesn't apply because they're circulating frequently, especially in winter for respiratory diseases in temperate climates, and they're infecting people despite a high level of population immunity, maybe because of antigenic variation, maybe because of, reduced memory, durability, or whatever, any number of things. But then there are some viruses like measles and poliovirus where there's a good correlation between seropositivity in a population and the amount of disease you get. I'm more comfortable with disease than circulation, actually, virus circulation. This idea that they're the same isn't correct. I always thought herd immunity applied to disease, but I could be wrong.
Yeah, I sort of thought that there always had to be a transmission piece to it because that's what you were actually stopping.
Well, that's how I teach it in virology. You're absolutely right. But I have this gut feeling that that's not right because we don't really... How do we measure transmission before PCR?
Yeah,
we look at disease, right?
Well, and I think that we sometimes said infection, even though we weren't distinguishing transmission, disease, and infection.
No, I've become more sensitive in teaching now more recently. I mean, the beginning of my virology, I said we're stopping infection. Yeah, but that's... Me too. I don't do that anymore. And I do think it was a disease thing. And you assume that if there's no disease, you're not getting infected. But that assumes there's no asymptomatic infection. So it's a tough concept. I don't think it's straightforward.
I think that's their point.
Okay. I mean,
it's a tough concept.
I'm struggling with that. Since COVID, I've struggled with herd immunity, even in understanding it for other viruses, because we simply didn't have the tools to fit the definition, right? Stopping circulation. So I have a problem with it. But I think you get to a certain level of some kind of immunity, it has an impact on disease. That makes sense to me. I think I've just confused people more than...
This is an easy read. It's open access. It's an easy read. Anybody can read it. All the concepts are there. And it's something you can chew on yourself.
Just about 2 pages long.
Yes, it's quite straightforward. All right, now we go. Now the article is actually more straightforward in my mind. Well, actually, maybe not as much. It's a PLOS Pathogens paper. Smallpox vaccination induces a substantial increase in commensal skin bacteria that promote pathology and influence the host response. So this comes from a group, the University of Cambridge and the University of Bern and the Sanger Institute in the UK. First author is Evgenia Shmileva, and the last author is Jeffrey Smith.
It almost looks like they have kind of both last authors, Brian Ferguson and Jeffrey Smith, are asterisks.
It has asterisks, yes.
As if you could communicate with them both. So they're like 2 senior authors.
Rich, they have this author summary, and there's one sentence in there. All right, this is about smallpox which is caused by virus, and of course, vaccinia virus vaccines were used to eradicate it. They say, Rich, eradication was declared in 1980 without an understanding of the immunological correlates of protection. Isn't that cool?
We still don't really understand the immunological correlates of protection. That's a difficult thing to get your hands around. In particular, with a virus like this, where you're talking a couple of hundred genes, half of which are involved in immune evasion.
Yes, and that plays some role in this. So, as you know, the vaccine is given to people by scarification, right? A bifurcated needle with a droplet of vaccine is scraped into your shoulder, in the case of us. And they note that, you have bacteria on your skin, so that could introduce bacteria into the vaccination site. I don't know. I remember, do they wipe it with ethanol before I guess they do, right?
I think so. But I would imagine that was not necessarily always the case, especially in the field.
Well, and I think so too, but I'm not sure that completely sterilizes the location.
No, it's more of a show thing, right?
Right.
Go ahead.
Well, and also, as we're going to talk about, they said that early vaccine batches were grown on the flanks of animals.
Yes.
And so they weren't, bacteriologically sterile by any means. Then subsequently that changed, right, Rich?
Yes. Well, actually, throughout the smallpox eradication campaign, I believe that the viruses that were used were grown on animals. Okay. I don't think there was either chick or cell culture virus until second generation viruses like Acambus 2000. Okay. So Dryvax, I think, was grown on calves. And although this particular paper is not about bacteria that were in the vaccine, it's about commensal bacteria that are in the vaccinated individual. Nevertheless, the notion that there was bacteria in the vaccine, I think, probably plays into this. And the first thing that triggered in my mind was the old 1915 paper by F.W. Twort on the discovery of bacteriophage. Because what he was actually trying to do was figure out what vaccinia was. What is it that's in the vaccine? And you could take that vaccine grown on animals and streak it out, and a bunch of bacteria would grow out. Okay. And in the process, he noticed that some of those bacteria got trashed. They basically got plaques. If you read, I read the paper closely again. It never ceases to amaze me. And he could propagate whatever this agent was that killed these bacteria. So it was a huge rabbit hole for him. Okay. But the point is that the smallpox vaccine was loaded with bacteria.
I presume the contemporary preparations were not full of bacteria, right?
What do you mean by contemporary?
In the last 20 years, they're grown in cell culture.
They're
and
grown
sterile,
in cell culture,
right?
yes.
Yeah.
Yeah. What passed for vaccine during a smallpox eradication campaign would, it would be horrifying to any regulatory person nowadays.
So my Acambus was probably sterile.
Your Acambus was sterile. That was a cell culture. That was a cell culture vaccine. They used to stick a little phenol. So the way you did this was to shave the belly of a calf, an anesthetized, obviously, calf. Shave the belly of the calf, and then they had this special tool that would you could load up with seed virus from a previously vaccinated animal and scarify in big long lines. Had about 5 different needles coming out of it. You could scarify the whole belly of the calf, so the calf basically got a confluent pustular lesion all over the belly. And then you would just drain this sort of after it developed, sort of scrape all the pus and stuff off the belly and collect it and mash it up and spin out the big chunks and add a little phenol to discourage further bacterial growth and then lyophilize it to distribute it. And that was the vaccine.
Notice he said pus.
Yeah.
So that sounds like what's mostly pus, Briam? Neutrophils?
Dead neutrophils.
Dead neutrophils. Wow. That's going to be relevant. So they noticed that even though there have been some, a number of side effects of the vaccination, none of them seem to involve bacterial infections. Even though, the early smallpox vaccine probably had bacteria, your skin has bacteria. So it doesn't seem to be a complication of infection. But they wanted to know if bacteria make a difference because they're there, right? And we know that bacteria and viruses are both present, and sometimes viral infection gives rise to bacterial infections, and viral infections can immunosuppress, et cetera. So they wanted to just know. It's kind of like Dick Moyer's paper that we did, Rich, where he wanted to know if scarifying versus injecting made a difference with this vaccine.
I'm glad you brought that up because that was the other thing that I thought about. And in fact, it's an important distinction to make in this paper, because in this paper, the technique that they are using to administer the vaccine is intradermal injection. And they're using a mouse model. So they inject a little bit of virus under the epidermis into the dermis. Okay, so above the basement membrane of the dermis. So that's intradermal, and that's different than scarification, where I've even got, I wonder if I've got a bifurcated needle here.
Oh, cool.
Yeah. Nice.
Nice. Very good.
Is it focusing on it?
Not quite.
It's hard for me to see.
Yeah. Yeah, truly a nerd who has a bifurcated needle right near his computer.
It's good.
But so scarification was this, the bifurcated needle has 2 points on it and a groove in between, and the groove holds a couple of microliters of solution that contains virus, and you just touch that to the skin, and the solution then by capillary action goes off onto the skin, and then you poke through that. The little points of the bifurcated needle, the 2 points, are sort of flared out so it can't penetrate too deeply. And you just poke it about 25 times until you draw a little blood. So, and the Moyer paper that you mentioned, and we can actually, I put this into the notes. By the way, the Tort paper that I mentioned before was another invincible TWiV.
Yeah, that's right.
It was the original invincible TWiV. It was me and Alan Dove talking about CC Tort or FW Tort's paper, TWiV 145, back in the Stone Age. And then there's TWiV 284. By the pricking of my thumbs, something wicked this way comes. And this is about Moyer's paper, which showed that if you just do an intradermal injection of vaccinia virus in rabbits,
yeah,
you get that'll kill them. If you scarify them with the same virus, they'll get a reaction and survive. But then, if you do an intradermal injection and then do a mock scarification immediately afterwards by pricking the skin all around the intradermal injection site, okay, they survive as if they were vaccinated. So the very act of annoying the epidermis with a bifurcated needle affects the immune response, okay? Their interpretation was that this turns on dendritic cells in particular, okay?
Yeah. Yep.
But so it doesn't really impact this paper. It came to mind immediately when I started reading the paper. But in this particular case, they're doing intradermal injections because that's the controlled way to do that in this particular model, and not scarification.
I have one question though about that. I don't know the answer. I'm sitting here trying to Google and I can't really find it. So they're specifically looking at the ear pinna of mice, which is quite thin.
Yeah. So what is the dermis like?
Yeah, exactly.
Is that your question?
Yeah.
Because is there even any muscle under there?
Right, exactly. Does the ear pinna kind of count as having all of these sections?
I don't know. Actually, they do have histological sections here. Have to look at those. See if they look like.
There's certainly cartilage, right? Just skin and cartilage.
There's something here that looks. Actually, if you look at Figure 4, the histology slides. I'm no histologist, but I have spent some time thinking about and talking about epidermis. I see something here that looks quite like an epidermis with even something that looks like muscle underneath it.
Okay.
A little bit.
I am definitely not a histologist, so I take that as being very useful information.
And this is also open access because it's in PLOS, so people can check out these figures.
There's something else under there, too. I mean, part of this, you're looking at a cross-section through the whole ear. There's something that looks quite vascularized.
Yes.
And I don't know what that is. Maybe I don't know what cartilage looks like. And there's got to be cartilage in here somewhere. At any rate, there you go.
So Rich, they use intradermal infection of the ear, and they say this causes a skin lesion that mimics smallpox vaccination in humans. And you just told us it's very different from scarification, right?
Well, in Dick's model, in both cases, you get a lesion.
Yeah.
In Dick's model, the subcutaneous infection was not well controlled. So the reaction was, not well controlled, and it caused a significant amount of lethality. And scarification, it was fairly well controlled. But my guess is, I'd have to look back at the paper, but I think if you took pictures throughout the progress of that, the lesions at the site of inoculation, whether it was scarification or intradermal inoculation, would initially look the same or similar.
Okay. All right. In that sense, then it's mimicking it. Yeah.
Okay. At the risk of taking us off on another tangent, I had this question come up very soon after what we've already discussed in my notes. What's the normal route of infection for smallpox or variola or vaccinia or something? Is any fraction of it by like rubbing skin with pox, or is it mostly respiratory? Or so in other words, how do these inoculation mechanisms relate to the natural way that people got infected?
For smallpox, it's respiratory. Okay. And in fact, the early attempts at artificial immunization involved inoculation, as we've discussed, which is where you take some smallpox material from lesions of a smallpox victim and introduce that intradermally. And in fact, I think that was, you introduce it in a scratch. And it can be a fairly deep scratch or a fairly shallow scratch, and I think that probably influences the outcome. But the case fatality rate from that procedure was a lot less than smallpox, and that probably is telling you, among other things, that the route of infection dramatically affects the course of the disease. Cowpox, I think in nature mostly is transmitted mechanically. Okay? But I'm not, at least in terms of human experience with it, okay? Humans pick up cowpox from animals, and that's virtually always in a fresh cut on the skin or something like that. But now the natural host of a lot of these pox viruses, like cowpox, is rodents. And to tell you the truth, I don't know how they pass it around.
All right, so they infect these mice intradermally in the ear, and they get a lesion at 6 days after infection, increases in size at 10 days, and it resolves in 3 weeks. He said this is a system that's been used to study viral virulence and immunogenicity. So they do this, and then they ask what cells are coming into the lesion. So they want to look at innate and cellular responses to vaccination. And what they do is show by flow cytometry an increase in neutrophils in the ear tissue at all times post-infection. And these, of course, neutrophils are important for bacterial infections. And they said, this not viral infections, mostly bacterial. And they said, maybe there's something going on with bacteria here. So let's take a look at that. Is that cool, Brianne?
That is absolutely cool. That is exactly kind of what I think of when I see neutrophils. Bacteria or maybe fungal infection, but usually bacterial infection.
Okay. So neutrophils are smoke. Bacteria is fire. Where there's smoke, there's fire.
I guess. I usually think of neutrophils as Pac-Man. And by doing phagocytosis and internalizing, that's very useful for getting rid of bacteria. But for viruses, it may be less so because you're also then letting them into a cell. And so I guess I would say it's more about where there's ghosts, there's Pac-Man.
One of the things neutrophils do is netosis, right,
Yes.
Brianne?
Yes.
And we did a paper on immune where they showed that it played a role in influenza infection even.
So neutrophils have this ability when they die to expel their chromatin as a net to trap extracellular bacteria.
So this is like falling on a hand grenade or something like that. It's amazing. Just in their death throes.
But for flu, that seems that, and I think antibodies involved in net-mediated inactivation of influenza virus.
Sure.
Anyway, so they may play some role. Anyway, so they said, are bacteria expanding? So they quantify colony-forming units of bacteria in the infected tissue. And in the vaccine-infected tissue, 100-fold greater than Mach at all time points. And 10 days post-infection, 1,000-fold greater. And what they put on was sterile, right? The virus is sterile. So they say, must be skin bacteria that are growing out after we put the virus on.
It's pretty impressive.
Yeah.
Yeah, it really is. So would the conclusion potentially be here that the vaccinia is immunosuppressing and allowing the bacteria to sort of grow? Is that
could be
that the idea?
That's one of their possible explanations. Yes, or at least part of the explanation.
And since this lab has all the gene deletions of all the antagonists, they can go through each one and check them out, right?
Yep.
And I'm sure they're doing it right now. That's a gold mine, right? So the bacteria growing out. They say we're probably underestimating the number of bacteria. So the bacteria and lesion size kind of peak at the same time. So maybe there's a link. All right. So what bacteria are in there? So to do this, you sequence the 16S ribosomal RNA. It's really nice that you can identify bacteria by doing a short piece of sequence. You can't do that with viruses. So they take the bacteria from the skin and the lesions, they extract DNA, sequence a specific part of 16S ribosomal RNA, and you can tell exactly what bacteria are there, at least what families. And they found bacteria from 4 families. And, listeners of TWiV may know some of these firmicutes, like Lactobacilli, Streptococci, Clostridia, Bacilli, Actinobacteria, Proteobacteria, Neisseria, Burkholderia, Bacteroidetes, and this is all part of the skin microbiota. All these family members are part of the skin microbiota of mice, which has been characterized, as you can imagine, by others. So it's from the skin, and not, as they say, not from the environment. They also see changes in the composition. So the microbiota, right, is a complex assembly of different bacterial species, and they see changes in that. By the way, mock infection itself, does modify the microbiome, right? So just, I don't know, they've been injecting saline, I guess. That process itself does it, which is interesting, right? I guess you're introducing some bacteria below the epidermis. And they go through some of the specific kinds of bacteria that go up and go down. And they say there's no particular type of bacterium that is preferentially amplified after vaccinia virus infection. Random bacteria in different samples. So the infection leads, and this is what I like about this paper, they summarize every paragraph. Thank you, Jeff, and your colleagues. Vaccinia infection leads to changes in the skin microbiome and expansion of opportunistic bacteria.
This is opportunism in spades, because it's almost like who gets there first gets amplified.
Yeah.
Because it's not the same every time.
Yeah. All right, so then they say, what's the relationship between the amplification of the bacteria and the lesion formation? So they give the mice an antibiotic, a broad spectrum antibiotic. They give it intraperitoneally for 13 days after infection. All right, so they have SPF, specific pathogen-free mice. They're infected with vaccinia. One group gets antibiotics, and another gets saline. And of course, when you give the mice antibiotics, you see a reduction in the lesion. No, I shouldn't say. Of course, that's the question. When you give the mice antibiotics, you see a reduction in lesion size.
So, could you define? They're going to talk about germ-free mice here in a bit as well. Could you please define for me specific pathogen-free mice and germ-free mice?
Oh, okay. Well, specific do you know the answer, Kathy? Otherwise, I'm going to look it up.
Oh, I can give you some of the answer.
Yes, give me some of the answer.
So specific pathogen-free is basically the mice in your mouse facility. So what you've got in your mouse facility in sort of a standard lab is that you have certain protections for keeping microbes out based on, the gloves and the gown and all the things that you're wearing. And you have some sentinel mice who are kept in the same room as your mice and who are tested from time to time for a list of pathogens, the specific pathogens.
Right. Yeah.
And so you basically know, and if your sentinels come back positive, then you have to make some changes in the room. And so the idea is that the mice in that room are free of any of the pathogens that are on that list, that specific pathogen list.
And
so
I presume that specific pathogen list is pathogens that you'd prefer not to have in your mouse colony.
Yeah, exactly.
Yeah.
And pathogens that, are going to really be a problem in your mouse colony. But you are not looking at mice that have absolutely no microbes.
Great.
Right.
I just had to renew my animal use, one of my animal use training courses yesterday. And one of the questions, they talked about germ-free mice and SPF mice. And then one of the multiple choice questions was, what does SPF stand for? It was really obvious, because they had really goofy, wrong answers. But yeah.
And then germ-free is a special facility where the mice are actually reared there and born in the complete absence of any microbes. So they're in special isolators where the water has no microbes, the food has no microbes, the moms have no microbes, so the babies are born with no microbes, and there's nothing. And when you actually do some dissections on those mice, they look weird.
Yeah, they're not normal, right?
No, not at all.
So are they, but they're not necessarily all born by C-section?
I don't think they are all born by C-section, because once they're actually derived in the colony, because mom doesn't have any microbes to give to them. But I think the first generations are born by C-section in order to get them germ-free at the beginning.
So that's part of this experiment. They then say, okay, is this an effect of an off-target effect of the antibiotic. Maybe it's something else. So they take germ-free mice and infect them and show they have delayed lesion formation, smaller lesions as well, which would happen because they don't have bacteria, right? So,
and if I understood it correctly, they did those germ-free experiments in Switzerland and in a germ-free mouse facility. And so at some point they talk about the fact that, some of the differences they could possibly be due to the fact that they're in different facilities. Even if they're not, even if they're all in Cambridge, they're still going to be, the germ-free are going to be in a different facility from the SPF mice.
okay. What else? So, virus titers with or without antibiotic treatment. No difference. Despite, different lesion sizes, viral titers in the infected ear was not changed by the use of antibiotics in the mice. So they say antibiotic treatment has no impact on viral replication, which is good to know because that's what we say all the time. And lesion size is influenced by the presence of bacteria rather than virus titer. Then they did sections of the ear, which we talked about earlier, and had a look at them. And they're unusual sections because they're very thin and long, which is what you would expect from an ear. They see a lot of cellular infiltrate, signs of inflammation, necrosis at day 8. And by day 12, most of the ear tissue is necrotic. Maybe that's what you're seeing there, Rich, those bubbles. I guess those bubbles are present everywhere.
I'm looking at the sections in Part B, which to me are easiest to look at in terms of trying to understand whether it really looks like an epidermis. It's not, I don't know. I'm, like I said, not enough.
It's funny. Those are from germ-free mice. You know that? Yeah. So they look different from SPFs. Now, it is a different magnification, but I don't see any of those bubbles there.
Well, if you look at the A section, Roman numeral 1 and 2,
yeah.
I see. That
I see
river of vacuoles-like things.
Yeah, I see it.
Whatever that is.
A river of vacuoles. That's a good way to put it. It's a river.
But also that there is this big difference in the necrosis and in the inflammatory cells in those treated with antibiotics versus not.
That's it. They compare them, the antibiotic treatment, much less infiltration and mild tissue necrosis compared to to no antibiotic. So they say, the skin bacteria promote lesion development, and smaller lesions, no bacteria, reduced bacteria as a consequence of antibiotics, but no effect on virus titers. Very interesting. All right, so then they looked at the immune response in these antibiotic-treated mice, and they noticed less recruitment of immune cells from day 1 to 4, but then inflammatory monocytes go up on day 5, followed by large leukocyte infiltration at day 7. And the antibiotic-treated group had a reduction in the recruitment of multiple subpopulations of cells at 7 days. And by day 12, these cell populations declined, except for neutrophils in the no antibiotic group, which continued to increase. So they say there is a notable positive correlation between lesion size and numbers of infiltrating neutrophils or TCR alpha-beta T cells. A correlation that it didn't hold for any other cell type. So, brand TCR alpha-beta T cells, what are they looking at there?
So alpha-beta T cells are sort of your standard T cells. They're what you think of when you think of T cells. They are contrasting them with gamma delta T cells, which are a more innate-like T cells that are found in places like the skin.
So this includes CD4s and CD8s?
CD4s and CD8s are in the alpha-beta subset, yeah.
Okay. They also measured cytokines and chemokines at different times in the ear tissue of control in antibiotic-treated mice. There were reductions in a variety of these inflammatory mediators in the antibiotic-treated mice, which they say is consistent with smaller lesions and reduction in cellular infiltration. Also, similar changes comparing germ-free with specific pathogen-free mice. So they say the presence of skin microbiota accelerates immune cell recruitment and cytokine chemokine production in vaccinia-infected skin. Then they looked at the immune response, the adaptive immune response to infection. They measured vaccinia-specific CD8 T cells and anti-vaccinia neutralizing antibodies. Brian, do you remember how they measured the T cells? Are you looking at peptides?
I don't remember. Let me see. I think that they're actually looking at, they're just looking at absolute number.
Yeah, I didn't think it was any sort of peptide specific.
Because they call it vaccinia specific.
Yeah,
That's
they
why
do.
I'm asking.
It's probably a tetramer.
Okay.
Let me check.
Back on the chemokine cytokine results, that those graphs were just wild at first in figure 6A. So once I figured it out, it was like, oh, this is a really good, concise way to show this, but it took me a minute.
They use something like a tetramer. They use a dextramer, but basically a tetramer to show vaccinia-specific T cells.
Okay. So what they find is that the numbers of absolute numbers is similar in all three groups of animals. So splenic CD4 and CD8 and vaccinia-specific CD8, similar in all the animals. Neither in splenocytes, no differences in splenocytes and non-T cells in vaccinated and germ-free mice, but the levels of neutralizing antibodies were threefold lower in germ-free than in either group of SPF. So basically all the T cells were similar between the two groups and a threefold difference in antibody. However, they say all the mice were protected from disease after intranasal challenge with a lethal dose of virus. So the threefold lower levels of antibodies don't matter, I guess, in that sense.
Does that ring any bells?
It certainly does. It certainly does.
It does.
Oh my God.
I think it's sort of weird that they see a difference in antibody levels in the germ-free, but not in the antibiotic-treated versus not antibiotic-treated. That surprised me.
Yeah. But anyway, basically that the animals are protected even though they have fewer neutralizing antibodies.
And maybe it's the T cells, right?
Maybe.
Yeah, I thought that was good. Anyway, so this does not affect their capacity. So what is this? So germ-free mice have an impaired capacity to generate neutralizing antibodies, but they're still protected from challenge. Now, I had this sentence I read 5 times this morning, and I couldn't wrap my head around. Do you ever get one of those sentences where you can't figure out what they're saying? So here, maybe I'm sure one of you got it. These data do not distinguish between the deficiency of antibody-dependent immunity in germ-free animals per se. It's the per se that gets me, I think, and the decline in humoral immune responses due to a lack of stimulatory signals from microbiota. Someone explain that to me.
Sure. So what they are saying here is that they can't tell if the reason why they were seeing a difference in neutralizing antibodies was because they weren't getting the microbiota introduced when they did the injection because they're germ-free mice, or if germ-free mice just in general are messed up in terms of their antibody production.
Got it. See, per se always throws me. And it's the Brits that use per se all the time. You notice? Well, maybe Kathy does. Do you use per se, Kathy? No.
Not too much, no.
Okay.
Never.
Because I'm not clear about, but I know now I understand they're defective in the ability to.
Yeah. So are they generally defective, or are they just defective in this specific case?
So I guess you could recolonize them and do the experiment, right?
Yeah, and that's what they say. They tried to recolonize them
right,
with Staph aureus, but then
they got sick.
They got really sick and all messed up, and so they couldn't do that.
That paragraph read very much to me like a response to a reviewer's comment.
Yes, that's what I exactly was going to say. Possibly requested by reviewer 3.
Yes.
Yeah, I mean, you shouldn't do monocolonization in general, though, right?
Right.
Right. And they actually talk about it in the discussion. That's where I started to be able to understand that question. And in the margin, again, I wrote reviewer 3 experiment. However, whether bacterial infection per se or immune system activation by bacteria induced tissue damage is unknown. So if you just leave out the word per se, it's okay.
Okay.
Whether bacterial infection or immune system activation by bacteria induced tissue damage is unknown.
Okay. Thank you. So what's responsible for this? Why are the bacteria expanding? And I guess they're increasing neutrophil infiltration. They're increasing cytokine and chemokine production. But it doesn't seem to matter in terms of protection against infection, right? So what do you think? Is this just well, first of all, the first question I asked, why is this happening? Their idea is first, vaccinia is immunosuppressing to a certain degree. They also say vaccinia shuts off host protein synthesis, and maybe, the skin cells make a lot of antimicrobial things, and maybe that's it, right? Was there another idea, Rich? You said there were a couple of ideas.
No, those are the
two main ones.
Think.
Yeah. But I guess I'm not sure it matters, right? Because in the end, you get good immunity no matter whether you have bacteria or not, right?
In this model, yeah.
In this particular model, yeah.
Yeah, I'd like to see a little bit more on the antibody thing, just because that was really kind of cool to me. Because if it, say, does change the antibody levels, but doesn't change the T cells, that could be useful in future design of other types of immunization protocols.
Yeah.
To me, the whole discussion, just in general, emphasizes that the way a vaccine is administered is important.
Yeah.
Okay. And just because you think in this particular case, just because you think you're administering something that's sterile doesn't mean that there's not going to be a bacterial component to the reaction. And like you, Vincent, I'm waiting for, this is kind of a tangent, but like you, I'm waiting for the patch. The microneedle patch.
Yeah.
That goes back to the discussion of scarification. Okay. I want to see that tested head to head with, a traditional intramuscular injection. It probably has been done. Okay. With some vaccine or other, because that would be great.
Yeah. I think it's being done with influenza. We should find some of those papers and do one of them.
I've remember reading those papers from way back in grad school.
Yeah.
But when they said, this could have some consequences for other vaccinations, it's like, I immediately thought about microneedle patches and bacteria and
But for vaccinia, it doesn't seem to matter, right? Because they say you get lesions without bacteria. You get, so you don't need the bacteria for lesions. They just get bigger. But there doesn't seem to be an impact on the immune response, except for the threefold difference, which we're not sure is specific to the microbiome, right? Is that the message we're getting? I know I understand that it could have relevance for other vaccines, but for smallpox vaccine, the microbiome expansion doesn't seem to play much of a role in protection, at least in mice, as Rich said.
Not in this model,
not in mice. Yeah.
So, Brianne, does the presence of neutrophils necessarily mean bacteria, or will they, they'll invade to some extent in the absence of, will they respond to just a viral infection or? So
they'll respond whenever there are the right chemoattractant cytokines present. Okay. And so, there probably are some neutrophils coming in whenever there's some tissue damage. So, when you have that swollen ankle, there's probably some neutrophils coming in there because of the chemoattractant cytokines. So they're probably coming in a lot of places, but classically, They are thought of as being for bacteria, protection against bacteria, and classically the cytokines that relate to them are thought of as being downstream of innate immune receptors that are turned on by bacterial products.
Okay. Cool.
Are we going to talk about zombie dye?
Sure.
In their flow cytometry, they excluded dead cells by addition of zombie fixable viability dye.
Yes.
So this is a commercial dye that has different permeability in live cells and dead cells. So you can use it to tell them apart, but it's called zombie dye.
Yes. So there's a dye people have always used in the past for this called live dead. And zombie is just a much better name than live dead.
We did a paper recently. I don't think you were on, Kathy. It was zombie yellow was another, right?
Mm-hmm.
And
this is the one that comes up from BioLegend is zombie aqua.
Aqua, different colors. Cool. All right. Did you like that, Rich? I thought you would.
Yeah, I got all excited about that, and among other things, because it triggered the memories of F.W. Twort and the scarification paper, and had me thinking about hematopoiesis and everything.
Yeah,
just got me going. Plus, all you got to do is whisper smallpox in my ear, and
that does
You'll follow.
yeah, I'll follow you. That's right.
Any immunization is going to introduce some bacteria.
Yeah.
Right?
Yeah.
So intramuscular, even though they do the alcohol theater, I would call it theater, although it probably helps to some extent, right? Is going to introduce some bacteria into the muscle, and who knows what effect it might have, right?
Yep.
A little bit of a natural adjuvant, although here it doesn't seem to matter. Okay.
And then the Twort was really trying to figure out what was going on with smallpox, and he discovered phage.
Yeah.
Yeah. Wow.
I looked up the paper, and it's not available except for the front page. But I wrote my friend Bill Summers, who's so he's a science historian with a long history of phage and herpes virus. Okay. And has a particular interest in the history of phage. And I knew he'd have a copy of this. So he sent it to me, and I will send it to you because it's worth a read. It's only a couple of pages. And actually, let me, oh man, what did I do with it? It's in my email somewhere. I'll get it later. The closing, you go ahead. I'll look it up because I need to quote this directly. You keep, I'll come back to this.
Do you guys want to do a couple of email?
Yeah.
You're
Sure.
good? Okay. Brianne, can you take that first one?
As soon as I get to it, yes. Charles writes, "Hello, Twivers. 64 Fahrenheit and waiting for rain in Chapel Hill, North Carolina. It is nice at the moment. I have a question about vaccines and sterilizing immunity. I have no problem with the concept that vaccines do not provide sterilizing immunity for the long term. My question is, do the mRNA COVID-19 vaccines provide short-term, say 60 to 90 days sterilizing immunity? If the answer is yes, or maybe, would that sterilizing immunity prevent people from being seropositive to the SARS-CoV-2 N protein during the 60 to 90 days? If that answer is also yes or maybe, I have a suggestion for why the CDC's MMWR that was discussed on TWiV 895 showed that younger people had a higher seroprevalence than older people. Older people have a higher rate of vaccination. If vaccination prevents N seroconversion for 60 to 90 days, that could be the reason for the lower seroprevalence. Just fewer days when an infection would produce seroconversion in a vaccinated person. Thanks, Charles." That's a really interesting point. I don't know if the mRNA vaccines are providing sterilizing immunity short term. I think that there is some data that there is protection from infection from some of those vaccines in the short term, but I don't know how sterilizing it would be and whether it is in fact able to be sterilizing enough to prevent N protein. I'd have to look at some experiments.
So you remember the early data on the vaccines where they were looking for basically PCR positivity, right?
Right.
They were in the 90s. So I think they're pretty good in that short time frame.
Right.
So the question is, would that prevent you from getting an antibody? Probably would, right?
I guess.
But I don't know if 90 days is enough to explain the difference between the old and the young people, though, because after that period, everybody's going to get infected at some point, or more people. So the thing about that study is that it was a convenience sample from the CDC, right? So they used antibodies that they had. They didn't do a study. And so maybe that influenced. Daniel pointed this out, that could have influenced the percentages as well. Maybe different kinds of people, young, middle, older people give blood or did whatever that was and went for healthcare. I don't remember what the specific issue was, but I'm not sure it's a good sample. A convenient sample of blood specimens submitted for testing. So I don't know what convenience sampling might limit generalizability. So they had them for other reasons?
I think they're probably just people who happened to show up for healthcare.
Yeah. So that's a bias. I don't know. It's an interesting idea. Kathy, can you take the next one?
Yes. Zaki writes, "The road to hell is paved with good intentions. Thanks for all that you do. It's made the lockdowns bearable and kept my mind expanding in ways I didn't feel qualified as a layperson. Also took my mind off the sad events around the world where those less fortunate than us suffered difficulties beyond our understanding. Zaki."
Interesting name.
Yeah.
Cool. Very cool. Rich?
Let me start with going back to this tort paper that I said. The very last paragraph in this paper says, "I regret that financial considerations have prevented me carrying these researches to a definite conclusion, but I have indicated the lines along which others more fortunately situated can proceed."
That's funny. That's very good. I like that.
Sad.
It's a great paper. It's one of these things where you look back and you realize, matter of fact, I think he was dealing with a lysogen. I probably shouldn't give that away. But look through it, and he observes things that are clearly plaques. Okay, and he does great experiments with it, and he has terrific insights. He starts out by saying, that we know viruses now only by the diseases that they cause, but much the same as bacteria, and they had a lot more information on bacteria at the time. He says, I'm betting that there are a whole bunch of viruses that don't cause any disease. Okay. And
cool.
It goes off on a rap about that. At any rate, it's very insightful, very, very fun paper. Okay. Deborah writes, "Vincent, I'm a fan of TWiV and regularly listen to the podcast on my long dog walks, occasionally needing to stop, rewind, and hear something again." I imagine her stopping when she's walking her dog. I thought,
dog gets pissed off.
Okay. Yeah, literally. "I am a clinician, pediatric infectious diseases, and enjoy the clinical updates, but also the virology and immunology deep dives. I was listening last week to the April 28 episode, 894, Dinner with the Temperance Family, when at the end I heard you mention the book For Love of Enzymes by Arthur Kornberg. My dad, Bob Lehman, was Arthur's postdoc in St. Louis University of Washington when his Nobel Prize winning work was done. They then both moved with the rest of the department from St. Louis to Stanford, where they remained for many decades. My dad is also a lover of enzymes, and a few years later discovered the mechanism of DNA ligase." She gives a link. "My dad is still a member of Stanford's biochemistry department, and at age 97, still participates actively in departmental activities, even after giving up his lab many years ago. I have attached a photo of them taken in their later years at Stanford. My dad is on the left, and I have no idea the story behind the matching blue shirts. When Arthur died, my dad lost a dear friend. Arthur, who was an MD, not a PhD, interestingly, was also a good family friend and actively supported my journey to medical school, and I will always treasure that. You mentioned that he was married 3 times, and that is true. His first 2 wives predeceased him, which is quite sad, but we were always so happy that he found love and companionship throughout his life. His legacy continues. As you know, 2 of his sons are accomplished scientists. One of them, Roger, is also a Nobel Prize recipient. Father-son Nobel laureates indeed. All the best. Looking forward to the next TWiV podcast. Best, Deb. That's Deborah Lehman, professor of clinical pediatrics at UCLA. Also Dean of Student Affairs. And there's a delightful picture of Arthur Kornberg and Bob Lehman. Very cool. Very nice. Thank you.
I just want to make it clear the way you read it. They were at WashU in St. Louis,
okay.
not St. Louis University at WashU. Yeah.
Okay.
WashU. Worcester.
We have a picture that Bill sent in. It's a photo of a cat wearing a knitted thing that looks like a coronavirus. It's green with spikes. That's cool. The cat doesn't look very happy.
No, this might be an image, an episode image.
Yes, I was thinking the same thing.
This, and under it says, new COVID variant, Omicron, a catastrophe. Newly engineered variant gives CDC pause, PAWS, which is great.
Very good. The cat doesn't look real happy.
No, but it's a green coronavirus.
It is
it's
green.
Not the typical color.
Yeah.
I don't imagine many cats being happy with something like that on their head.
Yeah, probably didn't stay on for very long.
No.
Brianne, can you take the next one?
Sure. Ellen writes, hi, TWiV. I'm in Minnesota where it's cold, and so occasionally hear Dr. Osterholm on local news talking about the pandemic. Today, May 2nd, I heard him on this segment, and she gives a link, of a local show talking about problems COVID-19 could pose in the future. One he raised at about 7 minutes, 30 seconds was, quote, we know that with coronaviruses, if you have a little bit of antibody, not a lot or none at all, you're in trouble. If you have none at all, you'll get the disease. Hopefully not be too serious. If you have a lot of antibody, you may get the disease. Hopefully it'll be mild. But if you get that middle group where it's just a little bit of antibody, that sets off an immune response. This is just like dengue and hemorrhagic fever viruses, which are 30 to 40% of the time fatal. I have a couple of questions. First, I was under the impression that the fatality rate of dengue after prior infection was due to have been previously infected with a different type of dengue, not the quantity of antibody. Is this accurate? Second, does this seem at all likely to happen with SARS-CoV-2? Last, I'm not a science communicator or scientist, so I'm wondering what your thoughts are on saying this to the general public. It seems like it could make people very fearful about naturally declining antibody levels. Thank you, Ellen. That's a great question, Ellen, and a great point. So this is getting at the idea of antibody-dependent enhancement. And I haven't seen a ton of data in people for enhancement of disease, although there was a really interesting paper from Judy Lieberman's lab that indicated that antibodies that came from infection, but not antibodies that came from vaccination, could lead to a change in monocytes in culture. And I think Vincent and Amy did that one. I loved the title of that episode, The Bonfire of the Monocytes. And so I have seen that paper. I think it's a fascinating paper that kind of could hint at some of these effects in cells, but I have not seen data on this actually happening in a person or in an animal. I think there was one hint of it in a SARS-CoV-1 paper. And so part of this in dengue is being infected previously with a different type of dengue, but then it relates to having antibodies that are somewhat less well able to bind. So that's how that quantity of antibody and different type of dengue thing relate. And I don't know of any data on this actually happening in people with SARS-CoV-2 at this point.
What is he talking about? Levels of antibody?
I think this is, frankly, you've been very kind here, Brianne. I think this is off the wall. I know nothing. I know of no data to support this. I'm not omniscient, but I'm not, totally out of the loop either.
I know of that one paper, and I think it's really cool, but it's in some cell culture infection of monocytes.
It's mediated by FC receptors, and it's very clear, but it has nothing to do with a little bit of meat, in between it. Goldilocks business here. I don't know what he's talking about.
And there was a huge concern early on that there would be antibody-dependent enhancement.
Sure.
During the development of vaccines, and people had they were watching this very closely in the early stages of vaccine development because that could have been a big problem. And I would think that if there were a serious problem with any sort of antibody-dependent enhancement in SARS-CoV-2, we'd know about it, and I'm certainly not aware of it.
Yeah, and that story that is sort of out there actually distinguishes between infection-induced antibodies and vaccine-induced antibodies. So if anyone is hearing this and is worried about vaccine-induced antibodies, no.
We're very careful to point
and
that
Ellen,
out.
you may not be a scientist, but it's in your blood there somewhere.
It's good.
You're a critical thinker, right on the money.
Really
you
good.
had dengue right, and you were right to question this.
Yeah. Her question about saying this to the general public, where he says, this is just like dengue and hemorrhagic fever viruses, which are 30 to 40% of the time fatal. It's just off the wall, broad generalization, and doesn't match the data about these viruses.
And is there, I guess dengue can lead to hemorrhagic fever, but are there other examples with hemorrhagic fever viruses of this?
No, not ADE, no.
Dr. Osterholm can be a bit of an alarmist.
Oh, yeah. Yep.
So this is not uncharacteristic.
Yeah. All right. The last one is for Kathy.
Aiden writes, hi, the problem of the plurality of a data point is solved. Wittgenstein's Tractatus, Logico-Philosophicus, 4.128. Logical forms are without number. Hence, there are no preeminent numbers in logic, and hence there is no possibility of philosophical monism or dualism, etc. Since a point is infinitesimal, it is an abstraction and therefore without number. Data can still be plural and involved with it.
Whew, boy.
So I have to admit that when a previous listener wrote in and said something about data point, and you guys kind of both agreed, I had no idea what was wrong with saying data point.
My understanding of that was that we are constantly going on about how data is plural. Mm-hmm. And my understanding of that comment was that data is plural and point is singular. So data point would be a non sequitur.
Yeah, I disagree, because to me, whenever I worry about should data be plural, I just change it to results.
Yeah.
And I think you could talk about a results point. I don't have a problem with that.
Well, data point isn't going away. I can't see people talking about datum point. That's not going to happen.
No, not happening. But what is it? Well, he says that a point is an infinitesimal.
Yeah, this is out there.
I don't get it.
I think basically a point doesn't exist. Right?
I think I'm thinking the and is a typo.
I'm not sure.
Since a point is infinitesimal.
I'm not so sure. I think there may be a thing called an infinitesimal.
and
I'm
infinitesimal.
guessing.
Okay, maybe we need more clarification.
Anyway, this Tracticus Logico-Philosophicus, I had not heard of it. Had any of you? It's a book-length philosophical work which deals with the relationship between language and reality and aims to define the limit of science. Okay.
So, sorry, Vincent.
That's all I have to say. I've never read it, and it exists. So the listener is quoting from a true thing.
So there is a noun that is infinitesimal, and it is a mathematical term defined as an indefinitely small quantity, a value approaching zero. So I think the implication here is that a point is an infinitesimal. Oh, I cut it out here. Let's see.
Right. Which is the same as saying a point is infinitesimal,
right,
in my book.
Yeah, okay, fine. But in my mind, he says it's an abstraction. It basically doesn't exist because it approaches zero. So you can't really define a quantity to it. So data point is okay. This is really for another podcast.
Okay. Not just another episode, but another podcast.
No, a whole different podcast.
Okay.
Either that or a podcast that has chemicals involved.
Okay. Very good.
Maybe, Joe Rogan can talk about this.
All right, let's do some picks. Brianne, what do you have for us this week?
So I have something that was made by some fellows at University of Wisconsin-Madison, which is a coloring book called Bacteria and Me. And so this has a number of different bacteria in it, obviously, but a number of microbiologists are described. There are some viruses. There's a bacteria versus viruses section. There is Jenner in celebration of the smallpox that we talked about today. And it just is really kind of fun and talks about some of the kind of people you think of as famous microbiologists, but also some people who are less well known, a lot of different types of organisms. And it just seems like fun.
Huh.
Oh, very nice.
I also like there are these little interspersed pages. I don't totally know what they are, but it looks like they maybe asked some kids questions about microbiology and like, what color would bacteria be? And there are funny answers sprinkled throughout.
If you click on Joseph Lister, this thing is sitting on his head. It looks like a bad hat.
Yeah.
It's funny. Oh my gosh. No, this is cool.
This would be good. I have a friend who, when they're on NIH study section, they said the only way they can concentrate is to do coloring. And so, when they're listening to other things, otherwise they get distracted and they want to multitask and read their email or whatever. So they do coloring so they can
color
kind
this.
of coloring. Yeah.
That's very cool. I like that. Kathy, what do you have for us?
I picked something that's smarter 3D printing, and it came from the University of Michigan. And it describes how when you're scanning to do 3D printing, you have to stop every now and then because it can get too hot and you can get basically warping of what you're scanning, what you're printing from it. So they have this other way of sampling how you scan. It's called Smart Scan. And so you just scan from different quadrants, or not even quadrants. They're much smaller than that. Anyway, it's an article that explains it, but the short video is like worth 1,000 words, and it shows you how this new way of doing 3D printing will be better, and you'll get less warping.
Cool. 3D printing is going to take over the world.
Yeah.
Can make an airplane by 3D printing? Probably one day.
Probably.
Yeah.
That's
one
neat.
day.
What do you have for us, Rich?
I have I believe it's a BBC production that has been on public television. This is I think that there are probably 4 episodes, and I think this is the 4th. I've been unsuccessful, even with my PBS passport thing, in finding the whole series. But this is a YouTube video of what I believe is the most recent episode, and it's about restoring Big Ben. which has been going on since 2017. And anybody who's been to the UK, to London recently, will know what I'm talking about, because Big Ben has been enshrouded in scaffolding, top to bottom. And this is an incredible undertaking to basically empty the tower and strip out everything that's not right and take all the appropriate measurements and reconstruct it and put it back in. Okay. This episode in particular has a lot to do with the clock and the clock face. And it's interesting because in the course of taking it apart, they discover that the original coloration on the clock face was different. Okay. It had some really brilliant blue and a lot of gilding and stuff, and they've restored that. And they've now unveiled the new clock faces. So if you go back to London now, you can see the new clock faces. At any rate, it's a fantastic series. And this episode has enough review in it so that you get an idea of what's gone on before. Some of the characters are interesting. They have a couple of characters that take care of the clock. Okay. And this is their whole life. Okay?
Cool. Very cool.
It's very good. I recommend it. This is 46 minutes long. It's worthwhile.
Cool.
That reminds me of the restoration YouTube channel I picked a couple of weeks ago. How people take things that are all messed up and they fix them. It's just amazing. Just amazing. That guy does it with paintings. This is a little bit different, but same idea.
I just wanted to point out that the 3D printing video is only about a minute long. So you can check that out.
That's very quick. My pick is a book about Burnett, McFarlane Burnett, who was, of course, an Australian scientist and Nobel laureate, virologist, immunologist, a character, really a character. Not a character, maybe a very complicated individual. Anyway, so this is his biography by Christopher Sexton. It was written in 91 originally, I think. He died in 85. And this is an updated edition published in 1999. He was a giant of science, really a genius. Amazing discoveries in virology and immunology. He was the head of the Walter and Eliza Hall Institute down there. He got a Nobel Prize with Peter Medawar, and then became a statesman of science from Australia. He wrote 16 books concerned with the relationship between science and society. And so anyway, it's just an interesting life. Just all over the place. And I wanted to read you one little passage here where he also has pictures in it. He says, he's talking about the Nobel Prize. I have a very sympathetic attitude to the personality of Alfred Nobel. He was something of a misfit with a brilliant intelligence and an ailing body. And he basically says, that's kind of how I am. And so I identify with him, and I know what he was thinking. His aim was to honor and reward those who had been responsible for achievements. The prize was to be large enough to provide a modest independence for life. I believe the prizes have played a great part in establishing the current sense of academic values, but I wonder greatly whether they are the most appropriate means to the end that Nobel desired. I fancy I have sufficient temperamental resemblance to Alfred Nobel to guess how he would have suggested the allotment of such prizes today. And so he goes on and says how he thinks the prizes should be allotted. Not for science in any way, not for peace, none of that stuff. For example, for the man or woman whose work had done the most for the understanding of human genetics and ways by which that would be applied to maintain the genetic health of our species. So he's a little out there. Anyway, really interesting life. And Brianne, you know him, his immunology most likely, right?
I do. Yes, clonal selection, very important.
And he did some virus experiments as well. I think he did some vaccinia stuff, right, Rich?
Yes, he did.
Yeah. I think, yeah. Anyway, really good bio. Steve Goff gave it to me to read, and it's got his name in it, and he doesn't want it back. So here it is, the incubator. We have a listener pick from Swellen. Hey, Vincent and all the TWiV team, including Dr. Daniel. Wanted to pass along this epitope, of a great show we get on PBS down here in the ATL. The show is Your Fantastic Mind, and it's produced by the Emory Brain Health Center. This particular epitope aired last season, but it is probably still relevant and contains a lot of great info on long COVID and what some doctors and researchers at Emory are doing to help patients. One patient is even a doctor himself. Segment is only 15 minutes long, so it fits into even the busiest of days. The other half of the epitope is about a new class of budding neurosurgeons, and that's pretty interesting too, though not COVID related, so enjoy. And if you want more episodes, they're all out there on the web. While I love the weather down here, I hate the politics, but it's really nice to live near Emory, Georgia State, Georgia Southern, Georgia Tech, and the CDC. Your correspondent in Roswell, Swellen. Thanks, Swellen. And that will do it for TWiV 897. You can find the show notes at microbe.tv/TWiV. You can send your comments, questions, pics to twiv@microbe.tv. And if you enjoy our work, we'd love your financial support, microbe.tv/contribute. Kathy Spindler is at the University of Michigan in Ann Arbor. Thanks, Kathy.
Thanks. This is a lot of fun.
Rich Conda is an emeritus professor, University of Florida, Gainesville, currently in Austin, Texas. Thank you, Rich.
Sure enough. Always a good time.
Brianne Barker's at Drew University, BioProf Barker on Twitter. Thanks, Brianne.
Thanks. It was great to be here.
I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV and Ronald Jenkins for the music. You've been listening to This Week in Virology. Thanks for joining us. We'll be back soon. Another TWiV is viral.